So I assume you want to learn about the cell. Well, you've come to the right place. Through this tour, we will show you numerous organelles and describe their function. So sit back, relax, and enjoy. Welcome to the nucleus. The nucleus is the control center of the cell. It contains the genetic information known as DNA, or deoxyribonucleic acid. It is a membrane-bound organelle, and at its simplest level, it is considered the brain of the cell. The type of cell is determined by that important DNA. DNA is also responsible for the creation of proteins and enzymes in cell functions. The membrane of the nucleus contains a phospholipid bilayer, which we will talk about later. It also has tiny pores, which are made of proteins. This is important for transcription and translation. The pores allow for charged materials to move into the nucleus. Nucleotides are a great example of material moving into the nucleus, and they help with the creation of messenger RNA, which can move out of the cell through the pores and help make proteins. Go.
On to the nucleolus. The nucleolus is where the DNA that is used to make ribosomes is found in the cell. The nucleolus is filled with fun proteins, and these proteins are used to synthesize the different types of ribonucleic acid molecules. Ribosomal RNA is one kind. It moves out of the nucleolus to combine with ribosomes, which produce proteins. These proteins will then move back into the nucleolus to assemble ribosomes.
Let me now introduce you to the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is a membrane which extends from the nucleus. It is important in the synthesis of lipids, phospholipids, and steroids. It also aids in breaking down carbohydrates. In blood, it helps rid the blood cells of unwanted toxic material. Good news for alcoholics. Alcoholics normally have a bunch of smooth ER, and it is what helps clean them out after a good night of drinking. Shame, shame. Muscle cells also contain a lot of smooth ER, because they are incredibly effective in containing calcium with a +2 charge. This type of calcium is crucial for muscle contractions.
Welcome to the rough endoplasmic reticulum. The rough endoplasmic reticulum is a folded membrane which is also extended from the nucleus. Unlike the smooth endoplasmic reticulum, ribosomes are attached to the outside of the rough endoplasmic reticulum, giving it the name rough ER. The rough ER is essentially the factory of the cell. It is crucial to protein synthesis. Inside the rough endoplasmic reticulum, proteins are folded to provide the structure needed for their individual functions.
Here's something fun to say. Golgi apparatus. The Golgi apparatus is a membrane-bound structure. It is composed of cisternae, which are essentially depressed tubes. Proteins which are created in a rough endoplasmic reticulum, we went there last time, will be packaged in transport vehicles and moved to the Golgi apparatus. The Golgi apparatus then modifies the proteins. Sugars are attached to proteins which create glycoproteins. The Golgi apparatus proceeds to load the modified proteins into little cell trucks and ship them out. It is essentially the monopolized shipping company of the cell. That's one monopoly we don't want to break up.
Now hold on to your horses, because this is the mitochondria. It is essentially the power plant of the cell, just as the V8 engine in the 1967 Shelby Mustang. The mitochondria is an individual cell which is found inside other cells. It has a membrane, DNA, ribosomes, and enzymes. It can also reproduce through binary fission. Inside the mitochondria's membrane, another folded membrane can be found in the form of cristae. It is like membraneception. Am I right? The cristae increases the surface area of the mitochondria. But as I mentioned before, the mitochondria produces the energy for the cell through a process known as cellular respiration. Cellular respiration produces ATP, which is what the cell uses for energy.
Now on to the chloroplast. Chloroplasts are not a part of animal cells, but they are a crucial part of plant and algae cells. They are very important to the process of converting light into sugar, which is called photosynthesis. Just like the mitochondria, the chloroplast is a membrane-bound structure which has its own DNA, ribosomes, and enzymes. It can also reproduce through binary fission. Chloroplasts contain the plastid chlorophyll, which makes plants green. Inside the chloroplast, thylakoids are found in a stat called grana. Stroma takes up the rest of the space between the grana and the membrane. The light reactions which occur in photosynthesis happen in the thylakoids. This reaction produces oxygen and glucose. The glucose is then used in ATP to fuel the plant cells.
Next are the vacuoles. Vacuoles are mostly found in plants. They can be found in animals, but they will be very small. If they are found in animals, they are used in endo and exocytosis. Vacuoles are important because they store water in the cell. Plant cells have very large vacuoles, and vacuoles are important because they maintain balance in turgor pressure. Proper turgor pressure ensures that the cell is properly inflated. Think about a plant. When you don't water a plant, what happens? It wilts, right? Well, that is because the water in the vacuole is decreasing, causing the cells to not be fully inflated. Once the vacuoles receive water, the cells will expand once again.
On to the lysosomes. Lysosomes are essentially the garbage disposal of the cell. Yuck. It contains digestive enzymes, which can break down any material the cell needs. This is called autophagy. It is also a membrane-bound structure, and thank goodness it is. If the membrane wasn't there, the digestive enzymes would seep into the cell and break it down, causing the cell to die. Sometimes the lysosome will pop and kill the cell. This is why the lysosome has earned the nickname suicide sac. The process of cell killing itself is known as apoptosis.
Next is the cytoskeleton. The cytoskeleton is essentially the cell's skeleton. It provides structure to the cell and helps to protect it. It is also important for cell motility and movement. There are 3 different structures which make up the cytoskeleton. There are microtubules, microfilaments, and intermediate filaments. Microtubules are the largest of the 3 and are essentially a big hollow tube. Their purpose is to provide support and aid in the movement of the cell. They can be centrosomes, spindle fibers, cilia, or flagella. Intermediate filaments are the mid-sized cytoskeleton components. They are permanent rods and will remain even after the cell dies. The main purpose is to reinforce and support the large microtubules. The smallest structures are the microfilaments. They are solid rods which provide a pulling force. The microfilaments are abundant in muscle tissue.
Don't go to sleep because next comes the centrioles. The centriole is part of the centrosome. The centriole controls where the nucleus and other parts of the cell are located in the cell. It achieves this through its positioning. It also helps with cell division by helping create the spindle which attaches the chromosomes and pulls them apart. The centriole is essentially the cell overseer.
Now we're at the end of this fine and dandy tour. We exit through the cell membrane, which is the structure which separates the inside of the cell from the outside. But it is selectively permeable. The cell membrane is made up of phospholipids, which are amphipathic. This means that they are hydrophobic and hydrophilic. The head of the phospholipid is hydrophilic, and the tails, well, they are hydrophobic. This causes an effective barrier where only tiny and or uncharged particles can get through. Large or charged particles can enter the cell through integral proteins. Proteins also act as enzymes, receptors, intracellular junctions, and attachment points for the cytoskeleton and extracellular matrix in the membrane. Cholesterol is also part of the cell membrane and helps keep it flexible.